my pleasure to be here, and I do get the honor of introducing Dave. Colluded sounds a little bit evil, doesn't it, Dave? But we have colluded for many years now. I mean, I first met Dave, actually it was prior to the chicken genome publication, 2004 and 2003 at Hickston. And actually prior to that, when I was with Monsanto, when sheep, the Dolly phenomena with the cloning of the sheep. So that's when I first met Dave, and we've been friends and collaborators for many years now. And as Rusty said, our goal has always been to try to improve the chicken genome has been our major focus, but avian genomics in general. And Dave has done a fantastic job over the years of really bringing a lot of folks together and promoting genomics in a general sense, and also the biology, of course, and how we interpret all that information and data mining. So rather than say a whole lot of things about the past, Dave, it's my pleasure to introduce Dave Burt. I think he's going to give us a wonderful tour of the past here, starting with the publication of the chicken genome. So welcome, Dave.
Good. Thank you. Okay. So we get this working first. Okay, thank you very much, Wes, for that very nice introduction. Could I have the lights down a bit? That's it, perfect. Right. Okay. Okay, well, it's a great pleasure to be here to start the Avian Model Systems meeting up, and particularly it's a great privilege to talk about and celebrate 10 years since we published a number of papers on the chicken genome sequence. And it's also quite fitting to do this at Cold Spring Harbor because just a few months after we published this paper, we had one of the first, at that time it was chicken genome, but then mutated into the Avian Model Systems meeting. We had a very nice meeting at Cold Spring Harbor in this very hall. So we appeared about 3 or 4 days. We talked about what did we learn from having this genome sequence with a host of speakers. And it's nice to see that in this meeting, still some of them survive. Get this working here. And the chicken, at that time, it was sequenced for several reasons, but in particular, one of the obvious ones was it's a very important food source. But at the time, most interestingly, based on some earlier work, it was thought sequencing the chicken and a number of other, what we could call them anchor species, would help us to understand, on the one hand, the constraints, the evolution of the human genome, but on the other hand, would actually set us up for the future for looking at the evolution of all these different genomes. And as you see as I go through the talk, I think that's happened, and today, we have so many genomes, we don't know what to do with them all. And like Wes said, many of us actually met at a meeting. I can't remember exactly. I think this was about a few months probably after Wes finished the first draft of the chicken genome sequence. And we all got together at Hingston, a little small band of people you recognize here, to get together and sort of think, well, what are we going to do with this sequence? And during the following year, we were working furiously on every aspect we could think of to analyze this sequence. And we ended up coming up with, like I said, a whole bunch of papers. There's hundreds of people on it. But you'll recognize this is quite a nice, I like this picture because you can see Wes hasn't changed at all. And there's Olivia here, me, and I think Darren's creeping around in the back row as he usually does. And I think Parker Anton's in there as well. So it's quite nice to see everyone here today. So, you've seen Wes already. So this was a meeting we had a couple of years ago. But that first genome sequence was put together by sort of classic shotgun sequencing strategy, which worked very well the first time, using a lot of information. And that resulted in this sequence that we analyzed. And that gave us the first time some information, not only about the first livestock genome, but also about the first bird genome. And birds are strange. And this is some pictures that Darren gave me years ago. And it contrasts sort of typical mammalian karyotype with a bird, chicken. Also really colorful, which is why I really like it. So he's painted all the chromosomes to different colors, so you can see who's who, which is which. So these human chromosomes are nice and colorful, very similar in size, pretty boring in terms of their size distribution. And this is not so colorful, but it's one of the early paint that Darren did. And you can see some of these large chromosomes, the so-called macrochromosomes, and loads, about 30 or so, tiny microchromosomes. And when we looked at the genome sequence, you could actually quite clearly divide them into two groups, just based on their properties. Things like GC composition, size of genes, size of spacing between genes, and recombination rates, the composition of repeats, things like that. So the macrochromosomes highlighted here tend to have larger exons, in fact larger intergenic DNA, so they're less compact. In contrast, the microchromosomes are much more compact and have fewer repeats. So it's almost like there are two domains in the genome, the macro and the micro domain. In fact, I don't have pictures here, but even when you look at their distribution in an interphase nucleus, they're in different compartments. Right. That sequence also, for the first time, allowed us to compare homologies between chickens and other vertebrates. In this case, in the paper, human and fugu, so fish and humans. And you can see in blue there a sort of core set of genes. And then in green, a set of homologs. And then in white, who knows, maybe species-specific genes. You could classify genes for the first time. And for interest for me, it's this sort of outlier of genes that evolve quite rapidly, and these tend to be genes involved in the innate immune system. And several years before, I published a paper on conservation of synteny between the chicken, human and mouse. And to my surprise, and everyone's surprise, we found particularly quite long segments of homology. But it wasn't until we had the genome sequence where we could actually say, yes, indeed, there were actually large regions that the composition of genes was the same in chicken as in human. But this early work really didn't look at the order of genes. That was going to come later. So those were some of the first glimpses of kind of properties of the chicken genome. And for the community out there, we were also interested in what was the quality like. Well, at that first time, there were some issues about sex chromosomes. They're all scrambled, and certain other parts were gaps and things like that. And Wes, as champion, and has been, like I say, apart from being a great friend, he's been a great friend to the chicken community by virtually every year taking advantage of new technologies. Obviously like 444 sequencing, Illumina sequencing, and you'll hear, I think on Friday, about some work he's been doing with PacBio. And at the same time, talking with the community, using all their BAC maps and their genetic linkage maps, trying to integrate all that information. And by so doing that, every couple of years or so, you would have another genome assembly, which was better and better and better, and integrated more and more into the resources. So there's always room for improvement. So this is just a quick picture of the karyotype. And in red are the problem areas. These one of them is this chromosome here, 16, which contains the MHC. And that's a classic area of a lot of gene duplications. And that's been very difficult to assemble. So hopefully in the next year or two, that's going to be assembled, probably BAC by BAC or clone by clone. Then there are these other chromosomes, these tiny, tiny microchromosomes, probably very, very GC rich. We don't actually have sequences assigned to those, even though probably we have a lot of the sequences in a pile in the assembly. So we need to assign those. And maybe the likes of Darren, well, once he gets the information, we'll be able to join the two things together. But there's certainly plenty of gaps, and let's at the bottom. And there are generally sort of three approaches that are being taken. One is using PacBio to try and fill in the gaps, and the other is using chromosome sorting, and the last one is optical maps to try and make effectively a restriction map of the chicken genome, and then order all the contigs, check everything's going, it's assembled properly, but at least to say this is the parts that are missing. So I won't say any more about that, because I'm sure that Wes on Friday will tell us a lot more about the latest in the genome assembly. So it didn't really stop there. The chicken genome, yes, was the first bird genome, but there were several more afterwards that we were involved with. First of all, David Clayton and his colleagues here, for example, in Wales, were involved in sequencing the zebrafinch genome. And then that was followed a few years later by the turkey. The turkey was quite interesting because that initially started with some fun, with ideas to do a pilot project, and then to obtain funds from the USDA and other sources to finish it. So we had a little whip round, if you know what that means. We looked in our consumable budgets and came up with about $10,000 each, and put that into the kitty to sequence the turkey genome as a pilot project. But because things were moving so rapidly, the group in the US actually sequenced the entire thing using 454 technology at the time. And then last year, we published in collaboration with BGI and groups in Beijing, the duck genome sequence. And that was entirely based on Illumina-based sequencing. And these are great because you can now, for the first time, start to compare between different birds. And you can see, yeah, use a sort of core set of genes here, common to all vertebrates. But there certainly were genes that appeared to only exist in birds. And similarly, only genes that existed in mammals. You could also start to look at, now we have a few more birds, you could start asking questions about gene gains and gene losses, which, again, is a very difficult question to ask. It's easy to say that I see a gene, but when a gene is missing, it could be for technical reasons, or it could be for more interesting biological reasons. But when you as you get more and more genomes, you get more and more confident that this gene is probably missing from this particular species or clade species. So this diagram here is just an overview, kind of give you, take all the genes and start to give you an idea of the rate of gain and loss of genes in gene families. And the take-home message from here is that birds basically have generally lost more genes than they've gained, and mammals the other way around. They've tended to gain more genes. So it's almost like the mammals are bloated and the chickens and birds are getting more and more compact. So it doesn't stop there. Over the last couple of years, a project led by Eric Jarvis, Goji Zhang, and Tom Gilbert have been leading the Avian Phylogenetics genomics project, involving, again, loads and loads of people, particularly those in museums and interested in bird evolution. And together, sequence another 45 avian genomes. So now we have, in this group anyway, a set of 48 that have been analyzed. And I did a count the other day, and I think there are about 62 or so avian genomes out there, ranging from a high-quality genome like the chicken to a lot of these more draft genomes that give you gene lists. So here's like the birds on the old orders, the galliforms and seriforms, and a couple of the ratites. And then this is a list of the other 44 neonaves, like songbirds, eagles, hawks, etc. All of these have been sequenced. And why is that? Well, one of the questions has been in phylogenetics. There's been a great interest for a long time. Has been the apparent rapid evolution of birds since the KT boundary of 65 million years ago. And it's been impossible to resolve this root of this branch of this tree. And Alan Baker over here coined this term the wall of death, because you couldn't actually resolve that phylogeny. And to give you a glimpse of this, to embarrass one of my PhD students, Veronica, she has a poster, I think it's number 41. So if you go along and have a chat with her, she'll show us some of her work on the CSF1 receptor and its ligand, but also it's based on using these 48 avian genomes. And if any of you have ever done any evolutionary study on gene trees and proteins, and you want to look at selection, particularly looking for evidence of positive selection at amino acid sites, you're always kind of scrabbling around in birds to try and get a few species. Well, here she's got 45 in this particular tree. So the minimum for these kind of analyses, for a perfect analysis, is about 10. So she's got plenty to do a very powerful analysis for positive selection using the various bioinformatic packages. So she can tell you a bit about that if you go and see her on Thursday afternoon. So all this information, a lot of it on genomes, and I'll come to it in a minute, annotations, genetic variants, have been summarized and are summarized in genome databases. And we, Roslin, particularly worked over the years, 10 years, with the Ensemble team, and now are within EBI at Hingston in Cambridge. And that's continued, and we currently have a grant, a joint grant between our groups to continue to annotate the chicken genome, as well as pigs, cow, and sheep, and salmon. But for these other bird genomes, we've created, using the same database system, 45 new Ensemble databases to make this all available. I've just described data available to everybody so they can look at that information. And Leo Eroy has done this. So the plan is in the springtime, when these other papers come out, that this will be made available as well. So you can see the papers and the data at the same time. And this will be made available through the National Avian Research Facility website, which is a BAC Wellcome Trust funded project in the UK. So at least it's got a long-term future, 5-10 years. So we can keep this information so you can see it and link it in with other biological resources, as well as the informatic resources. Richard Kuo has done this work, and you can contact him. And Alan Hart is at this meeting, and he'll be able to tell you a bit more about the NARF. This actually raises just creating these 45 databases. You can't keep doing this. The plans, this sounds crazy, but the plans are to actually sequence all the bird genomes, species that we can get our hands on. So I gather at the moment, through particularly the museums in Washington, Eric and his friends have got about 8,500 DNA samples. So in collaboration with BGI, all of these birds are being sequenced. So currently, we're up to about 200 birds. So in another year or so, there'll be another avalanche of sequences coming out. So that'll be an amazing resource, but also a major headache. How do you annotate? How do you manage all that data? It's a big question. So hopefully one of these days we'll be able to do it in parallel. That's the idea. So this brings me to the question of gene annotation, which will probably be a great interest to most of you here. It's also one of the biggest complaints, the biggest problems when any genome comes out. So again, just like the genome assembly, gene annotation has evolved over time. It's getting better and better. So this is the typical Ensemble pipeline of homology-based modeling of genes. And traditionally, it's focused on proteins. So you might get proteins that are already being sequenced in chicken, and find out where the genes are, and then work out the intron-extron structure. But more often than not, it's done by homology with other species, like humans, mice, fish, etc. But given an incomplete genome assembly, then you've got gaps and missing parts of the genome. You might actually have genes that are missing, or you might have genes that are fragmented, or we may not be sure if this gene is an allele, this gene. So you've got some problems like that. And this leads to incomplete models, missing genes. And the gene annotation process itself is not perfect, so you kind of miss things out. And one problem case will be rapidly evolving genes. So in this case, rapidly evolving proteins, like the CSF1 that I talked about earlier. It's quite difficult to use homology modeling to find these things. So the better the genome assembly is, the better the models are going to be. I should add at this point that when you look for genes in the genome, you can find them, you can find complete genes, and you quite often find bits of genes. And the ensemble pipeline is very conservative. So even if you can find half of a gene, it won't annotate it. So that's an issue. It's something we need to flag. But with a better genome assembly, we'll be able to put the two halves together and give you more genes. But in terms of annotation, one of the developments in only the last couple of years has been the introduction of new data from RNAseq. And many of you have used this technique for quantification and discovery of RNAs. And 2 years ago, we had a meeting in Edinburgh of the Avian Model Systems meeting. And in that meeting, we all agreed that we would try to pool our RNAseq resources. So this is the product of that. So about 13 different labs in the end provided data on over 100 libraries. I think it's about 120 now, from a whole range of different tissues under various conditions, males, females, infected, non-infected, things like that. So there's a real rich source of RNA-seq data that Ensemble were able to use to annotate the chicken genome. And they did that, I think, about 2 or 3 builds ago. And that's been quite good. So here's a comparison of before and after. This is one of the earlier, this is just an example of a zinc finger gene. Zinc fingers are a real pain to annotate because there's so many of them, and they're quite tricky to work exactly what the structure is. But the previous annotation was a bit sketchy. There's a couple exons here, there's one here, for example, and something broken here. And then down here we've got some EST data and some data from cDNAs. And you can see, well, there's something wrong here. Well, there's no evidence for this. And this definitely shouldn't be there. But in the new build with the RNA-seq data, you get a nice annotation. There's still something wrong at this end, that the model isn't going right out here. And the nice thing about the new Ensemble view is that you can look at the RNA-seq data here, some data from an embryo, and you can see all the RNA-seq data, and you can see it perfectly matches this exon, this one here, but then there's something missing in the end. So this clearly needs to be an extension of 3' UTR. Also, I'm not going to talk about it here because I don't have time, and the data is still in its still preliminary. The question of non-coding RNAs. And as you know, the last 5 years or so, we used to throw away all the RNAs, all the ESTs didn't quite fit, but most of those were non-coding RNAs, maybe regulatory RNAs. And we certainly, using homology methods, have been able to up the number of non-coding RNAs, especially the short ones, up to about 2,000 or so now. So that's been quite a big improvement. So a lot of microRNAs, for example. But there's also a class of interesting RNAs called long non-coding RNAs. And we have currently an estimate of about 15,000, at least. But depending, if we have 3 groups that work on the same data, they all come up with 3 different answers. So we have a situation where we're trying to sort out that kind of data. And what we're doing, we're using the 48 genomes, avian genomes, to try and sort that out. So again, hopefully in the next few months, that will be solved. So anyway, having these resources are amazing, but what I wanted to do is give you a quick example of one of the problems with rapidly evolving genes. And the innate immune genes, like I said at the beginning, are a typical example. And this is just a summary of all the kind of typical innate immune gene families you find. So the overall conclusion here is that generally birds have the same kind of sets of genes you find in mammals. But if you look down here, the number is about 2/3. And that's quite consistent. So birds probably do have fewer genes. But like I said in the beginning, birds tend to have fewer genes anyway. But when we have a gain or a loss of gene, we've got to be careful. It could be interesting, or it could just simply be an artifact. So here's a few examples of some genes that have been difficult to find, in fact, impossible to find until recently. And so maybe filling in the gaps, we'll start finding these things. We'll have one here that Veronica's been working on, which she'll tell you about her poster. But it's a nice example of be careful when you think a gene's missing. It's a CSF1 gene. And if we take a region in a chicken that is homologous to a region in human, we can see basically there's a gap that covers this homologous area. So there's a physical gap, there's a technical gap. Can we fill it? Well, what I did was looked in the zebrafinch genome, and I found in that homologous area a fragment of a gene for CSF1. Then we were able to use that as a probe to pull out a BAC clone. And we were able to find a good CSF1 gene, and then we were able to go back to the other birds and find them in those. And Veronica sequences BAC clone and has found the CSF1 gene and all these other genes. So all of these genes here are missing, supposedly, in the chicken genome. But you can find some of them in EST databases, but most of them are actually missing. So why is that? Who knows? Could be something to do with they just don't clone very well. They don't amplify by PCR very well. So I actually used here PCR-free Illumina sequencing. And we also use PacBio to sequence this clone as well. But anyway, it's there. So now she's got something to work on in terms of genes. But I think a lot of other genes that are missing will also be in this boat. So this is a list of the summary of the statistics of the gene annotation. Like I say, there's about 15,500 coding genes and about a couple thousand non-coding RNAs so far. And that list will grow. The nice thing about Avian RNAseq data is that you can also ask questions about, well, how many of these transcripts, how many of these genes are actually expressed in different tissues. So again, this list will grow. I hope to create over the next year an atlas. So you can look into this atlas and say, where's the genes expressed and in which tissue and things like that. And you can also look at the data and look at the exons quite nicely. So you can actually, for the first time, say, yes, that's a real gene, as opposed to some ab initio thing, so you can verify it. But RNA-seq data is really good at defining all these exons. It's not very good at stitching them together into transcripts. So assembling splice isoforms is, like I say, is very tricky. So what we've been doing very, very recently, this is only in the last couple of months, has been working with GATC as a partner to sequence and normalize libraries, libraries in the sense of synthetic libraries in a way in the test tube. Normalized libraries, and we sequence them using PacBio technology so we can get full-length cDNA sequences. And the idea then we can then use them to look for splice variants. So we use this template switching technique to get almost full-length cDNAs. And then a traditional normalization method with a few cycles. And you can see this before and after. And then we take a few size fractions, the smallish size around about kb, and a larger size about 1 to 3 kb. And then this is just a distribution of the cDNAs. You can see most of them are in about 2 kb or so, but there's some up here, up to about 7 kb. So this is very, very preliminary, like a few days ago. And what I'm showing here is just a random gene that we've picked. This is the ensemble exon intron structure. And this red line here is the PacBio cDNA sequence of this gene. And it goes right to the very end, 3' end, and right to the very beginning. So that one's a really good one. So hopefully using these cDNAs, sequences, these long reads, we have currently about 1.5 million long reads. We should be able to not only verify intron-exon structures, we should also be able to build better splice variants, which I think a lot of people would like to see. It is interesting to contrast how things have moved on. About 12 years ago or so, I think it was 2002, a group of us in the UK were part of an EST consortium in the chicken, the so-called BBSRC EST database. And that cost over £1 million for about 300,000 ESTs. This project here produced 1.5 million long cDNAs from a normalized library, sort of equivalent, so we get 5 times as much for about £20,000. So it's a lot less. So the plan over the next few months is do another 5 from different sources, and maybe go beyond that and use this as a way to sequence full-length transcripts and use the RNAseq data that I described earlier to quantify the different isoforms. So the other side of what's genome sequencing is genetic variation. So in the chicken world, genetic mapping is really important, obviously. It pays for a lot of the research. We want to discover genetic markers for traits like you see here, egg production, growth. And it also, particularly in the last few years, we've been concentrating on infectious diseases like avian influenza, Marek's disease, Eimeria, etc. So what we would like to be able to define are our genetic markers, genes, and even causal variants that underlie resistant to these many diseases. And this spawned a whole branch of area called QTL Mapping, which basically is an association between genetic markers and traits. And it's been very successful in all the farm species, and in chicken, I can't remember when this was done, just end of last year, there were about 4,000 QTLs. So 4,000 regions on the genome encode genes that control a few hundred different traits. So that's really good. However, when you look at those sort of results, this is a typical result. You get here's a chromosome, about half of it. We can say in there somewhere, there's a gene that controls this particular trait. And probably it's a gene maybe in here somewhere. But to get that resolution is quite difficult. And also that we can quite often only account for a small percentage of the genetic variation in any given trait. So there are probably more genes as well in the rest of the genome. And again, so this spawned this idea of we need more markers. What we would like to have are enough genetic markers to cover the entire genome. So there's sort of 2 branches things are moving. One is that we've done that now, and I'll show you that in a second. So hopefully we can use those technologies to get down to individual genes. For some of the geneticists, they say, I don't care about the genes anymore. Just give me all the markers and I can account for everything. So that's the genome-wide selection procedures. And that's been taken up by industry now, and that's going quite nicely. So what you really need there are markers. So again, in parallel with the genome sequence paper 10 years ago, we set out to sequence and find single nucleotide polymorphisms, SNPs, and we found 3 million, so that was quite good. And that was used in a series of experiments to make some low density markers, panels, and we were able to show that how they use those to look for SNPs and SNP associations in layers and broilers. And it was quite interesting. We found that there were these sort of blocks of linkage disequilibrium that were quite large in layers and very small in broilers. Unfortunately, the number of markers we had at the time weren't sufficient to map anything that's useful. But with the advent of next generation sequence technology, panels of higher density were being produced. This one by Martin Groningen. And then about a couple of years ago, we sequenced a few hundred birds using Illumina technology, detected 40 million putative SNPs. And from that, we created, with Affymetrix, a 600K array that's publicly available and being used all over the place to map traits. It's quite a large collaboration. Again, another one of these consortia. So SNPs, we find SNPs, large numbers, several million per in each of the different lines. This marker chip was constructed specifically to have markers distributed over the genome, not in terms of their physical distribution, but in terms of their genetic distribution. So in green, you can see it's fairly even when you consider the number of markers per centimorgan, and in red, it's not. And that's because on the microchromosomes, at this end, the recombination rate is very high, so you need more markers. So this is the ideal SNP chip, and it works everywhere. So these are a range of different commercial broiler and layer lines, and you can get at least 100, 150 informative markers. And for genome-wide selection and GWAS, you need about 100,000. And these are the types of applications that have happened over the last few years. So you can see a wide variety of things that you can use these chips for. This is a fun one I like to show. This is looking at using the SNP chip to look at population structures. So you can basically take a piece of meat out there, get some DNA, type it, and I can tell you what kind of chicken it is, you know, white egg layer or broiler. And if I've got the right samples, I can tell you which line within which company it comes from. So it's really amazing. But it's more interesting to go further, as far as I'm concerned, to look at are these SNPs and other genetic variants, can we actually predict whether they're doing anything in terms of are they functional? So it's quite important to look at those 40 million SNPs and say to yourself, are any of them artifacts? Because you know a lot of next generation sequencing technology is plagued by false positives. So we sequence in the good old-fashioned way 100 or so random fragments of DNA using Sanger sequencing to actually confirm by eye SNPs and indels and things like that. And using that as a sort of gold standard, we were able to come and look at the next generation reads and titre it basically and say, right, if we have a quality threshold at 40 and don't have any strand bias, we end up with 15 million SNPs, very high quality, that we know that less than 5% of them are false, and we're missing at that particular frequency, again, less than 5%. So looking at these SNPs, what kind of things can we do with them? Well, we can find out things like where do they map. That's easy enough. And if they're coding, we can look at in the coding regions to say, do they create a stop codon? Do they change an amino acid? So you can start to predict. If they create a stop codon, say the beginning of a gene, it's going to be a null. It's going to knock out the gene function completely. So it's quite amazing how many of these you actually do find segregating in the population. Most of those, of course, would be heterozygous, so they're not lethal, but some of them might be complete nulls, and the animal's living fine. So what about the future? Well, so far I've talked about genome assemblies briefly, multiple species, annotation, looking at transcripts, coding, non-coding, looking at genetic variations, variants. I haven't mentioned much about regulation. The transcript work is good, and it starts to tell us where splice signals are, where starts of transcription are, and the ends. So that's all towards starting to get at the regulatory regions. The constrained regions, they're going to be really important to define potential areas of regulation. And these are things that we're all interested here. But it's been a neglected area. So again, going back to that Nature paper, it was quite obvious just by looking at a whole range of different regulatory regions that they were certainly under constraint between humans and chickens. I know this paper came around about the same year by Kondo, using dot plots, basically look at highly conserved regions, and they're using these to predict where he thought enhancers were, and then making various constructs. And the chicken's brilliant for this, and then seeing the effect of those, in this case, in the SOX2 gene, and was able to assay for tissue-specific enhancers. But like I say, this is an area that's really been neglected in the bird, but now is the time to address this, I think. And you've all heard about the Human ENCODE project, and the idea behind this project, as it's developed, was to define a lot of the regulatory regions in the human genome. And all this information, kind of stuff I've talked about so far, gene annotation, transcription, but also a lot of the regulatory data, it's all pooled together and then made available to the community. So in January, in fact over last year, a group of us, again mostly in the livestock area, I've been colluding together, to say, scheming in, to try and come up with a project called agENCODE. And that is to do a kind of small version, an updated version of the Human ENCODE project for chicken, cattle, sheep, and salmon and pigs. And hopefully that will take off. So currently what's happening, groups around the world are, like I say, scheming, applying for grants to try and support such an initiative. The very least that will happen is that we'll continue to pool data, bring it all in, and Ensemble will be the place that you'll find a lot of this information. And we'll pool it together, and hopefully we'll have a better picture of the regulatory genome. Again, something we'll want to use. So if any of you are interested in doing this, or the RNAseq, or any of the other consortia, you can contact me, and if I can't help you, I can pass you on to somebody that will. So I think the agENCODE project brings it all together, and really great, because instead of just focused on human and mice, we can actually look at a whole range of vertebrates and start looking at the evolution not only of proteins, RNAs, but also the regulatory part of the genome. And this is an overused, but I quite like this. Theodore Jokanski's famous words, nothing in biology makes sense except in the light of evolution. It's very true, even more true today, I think. So I'd just like to finish off by acknowledging sets of consortia and many institutes around the world. And at the bottom, sets of funders that have funded me, as well as other people, to do this kind of work. And finish off with a picture I've used over and over and over given to me ages ago by Larry Coburn. I'm not sure if he's here, but he's certainly due to be here at this meeting. So thank you very much. Any questions?
It's open for questions. Oh, here, okay. Just wondering if you try to do a proof of concept on like identifying deleterious SNPs in animals and cross them and see that you get the expected mutant.
Yeah, not yet, but at Roslin we have an experimental population. We call it J-line, and we've sequenced all the birds in that population, and we find lots of SNPs, and that's exactly the type of thing we should be able to do next. Yeah. Richard?
On the commercial strain, so each year you have a selection, right? Do you have an idea about the proportion of SNP that change over some generation?
No, but that's a very good question. Yeah, I can say it. I have a project at the moment where we're looking at Marek's disease resistance in chickens. And this is with collaboration with Highline. And they've collected DNAs over a period of 15 years. So we'll be able to answer that question. Yeah. So it's good to collaborate with some of these companies because the number of animals and the resources they have. They were quite smart in that 20 years ago, 15, 20 years ago. A lot of them were thinking about marker-assisted selection and things like that. And they stashed away blood and things like that. So they thought maybe one day we'll be able to use it. And so days come. Okay, any more questions? No? Okay, so I'm here for the rest of the meeting. So I hope many of you want to be involved in some of these consortium. This is the spirit behind this meeting. So just come and talk to me.